All right, welcome everybody. I'm Forrest Meggers. I have the privilege today to introduce our speaker. I'm an assistant professor in the School of Architecture and at the Andlinger Center for Energy and Environment. Today we're very privileged to have Dr. Richard Sayre with us, the director of the Biofuels program at the New Mexico Consortium in conjunction with Los Alamos National Labs. And he's a renowned researcher in biofuels, as you can see and see from his CV. He's been involved in many programs regarding biofuels, but I don't want to go into the details. That you can read the extensive list of projects he's worked on online. And I think the most interesting thing that I found out, that since I was the person that gets the honor to introduce him, is that he did his PhD at Iowa and is from Iowa City. So I think the most important component to his background is that we both share a common thread, and he's then journeyed from there, doing his PhD at the University of Iowa to postdoc at Harvard. And I'm very excited to learn about making next generation biofuel systems work from his extensive background in this area. So thank you very much for coming, and I look forward to hearing your talk.
Thank you, Forrest. And thank you for the invitation to come to Princeton. This is my second visit, so I've gotten to know several of you through the Combustion Energy Frontier Research Center. Ed is actually my host, but Ed is not in town this week, so maybe there's a message there. I don't know. But anyway, it's nice to be back again. And so as Forrest was saying, I've been involved in a number of different projects. The project I'll be telling you about most extensively today related to biofuels is the National Alliance for Advanced Biofuels and Bioproducts. And that program was initially based at the Danforth Plant Science Center, where I was 3 years ago, and then more recently moved to Los Alamos National Lab. That was a DOE-funded program, a $70 million 3-year program in partnership with industry to look at the potential to produce biofuels from algae cradle to grave. So it included the algal biology, the cultivation, harvesting technologies, extraction, fuel conversion, which would be of perhaps greater interest to this audience, also co-products, and then to keep us all honest, life cycle and sustainability analyses by a team of economists and environmental scientists. And what I'll show you as we move along is that program developed a roadmap for where we need to go in the future to actually make this economically feasible as a biofuel technology system. But of course, we have to look at what are the competing technologies for producing energy in the US. And so what I've shown here is a ranking of energy technologies based on energy return on investment. And toward the top of the list, of course, is hydroelectric. But the extraction technologies based on fossil fuels are clearly among the highest for energy return on investment. And down at the bottom are more of the biological systems for renewable fuels production. Interestingly, there are actually numbers for food. We're not particularly good at converting fuel into food, but those are the numbers. Now, of course, one of the concerns that we have for various energy technologies is the carbon emissions associated with each of those technologies. And that's usually indexed by the carbon efficiency or amount of carbon dioxide released for each unit of energy produced. And of course, the extractive technologies do very poorly in that respect, whereas the renewable fuel technologies, and including those technologies in which carbon is fixed by photosynthesis, do much better. And so looking at the future of sustainable energy production, of course, we have to consider a mix of not only the energy return investment and carbon efficiency, but if the consumer is going to buy into the technology, of course, the economics of producing those various fuels and what the cost is for the consumer. So if we focus, and I'll focus now exclusively on biofuels for the remaining portion of the talk, as essentially a replacement for liquid fuels. And so biofuels, it's clearly recognized, will have their greatest impact on transportation, and in particular, perhaps aviation, which really only can operate on high energy density hydrocarbon-based fuels. And so there are a number of advantages, clearly, for the renewable systems. But perhaps the most important constraints really have to do with the fact that we're using solar energy ultimately to drive these biofuel production systems. And so how efficiently we actually use that solar energy is going to be a focus for a large part of the research that I'll be telling you about. So biocrude, and I'll show you some data on what biocrude is exactly in a moment, is a potential replacement for petroleum. It's a very hydrocarbon-rich fuel source, and it potentially can replace nearly all of these products coming from a barrel of petroleum. But perhaps most importantly is actually jet fuel. So we're seeing that cars, of course, are going to electric technologies. There are alternatives for a few of these other fuels, but diesel for heavy transport and also jet and aviation fuel, there really are no feasible alternatives other than hydrocarbon-based fuels. And that's largely for the reasons that are based here, and that is the high energy density of hydrocarbon-based fuels. But biofuels are making an impact on our global carbon emissions, and particularly in the United States. And so this data is some of the most recent from about 2009. And you can see that the biomass and biofuels contribution to the renewable fuels spectrum is appreciable. It's about 25% of the emission reductions up until 2009 could be attributed to biofuels. Now, of course, natural gas has made a huge impact because it's reducing the carbon emissions by a factor of 2 relative to coal. And of course, the cost of natural gas has dropped dramatically as well, associated, of course, with the increase in the supply. So if we're going to try to improve what we've done in the past with regard to producing biofuels, then we need to address primarily constraints centered around the sustainability issues. And very importantly, of course, as I noted earlier, is the energy return on investment, the amount of carbon that's released per unit energy. But where we live in the Southwest and New Mexico, of course, water is a major constraint. And so this is a facility in the southeast corner of New Mexico near the border with Texas. So this is oil country, New Mexico, and this shows a small-scale operation that's actually using produced water from oil wells, which is heavily contaminated with hydrocarbons and also salts as a medium to grow the algae. And so this water is being remediated as it's being used to grow algae. The algae are phase partitioning a large amount of the hydrocarbon residue remaining in the produced water, and so they're helping to clean it up. And there's the byproduct of producing fuel as a result of this. So this is a combined technology for mediating water as well as producing fuel. Of course, we're also concerned about competition for food, but one of the really attractive features about biofuels is that they're now compatible with all the existing infrastructure in the US for petroleum-based fuels. So UOP, General Electric, and the North Dakota Energy Efficiency Research Institute have all independently developed catalysts that can take biomass feedstock from almost any source now, but in particular oil-based algae, and convert that into the full spectrum of transportation fuels, diesel, gasoline, and aviation as well. And so the downstream technology has moved very rapidly in the last few years to make it possible to use biomass for producing liquid fuels. Scalability, and I'll address that in a moment, is a particular concern, and then, of course, ultimately is the economics. So there are a number of reasons why we think algae are particularly attractive as a biofuel system. And these advantages are shown here. But unlike plant photosynthetic systems, every cell, and these we call microalgae. We had a discussion earlier as I was visiting folks, what is a microalgae and what's a macroalgae? But these are single-celled organisms, about 10 microns in size. But every cell is photosynthetic, so there are no heterotrophic tissues that create a burden on the plant for that photosynthate that's being produced. They also have high-efficiency photosynthesis. We have a very active program in this area. I'm not going to talk about that today. But these algae use energy in the form of ATP to actively transport inorganic carbon into the cell, elevating the internal CO2 concentration, which saturates the enzyme that fixes CO2 and competitively inhibits a competing reaction called the oxygenase reaction. And that elevates the rate of photosynthesis by about 30%. So high-efficiency plants can do this as well, but they do it in a very different mechanism. It's called C4 photosynthesis. But in algae, it's essentially 4 genes. A very simple system to move the bicarbonate into the cell. We're actually now taking that system out of the algae and we put it into plants. And so we've re-engineered plants to have higher photosynthetic efficiency by taking advantage of the biochemistry that goes on in these cells. They have very high potential oil content, and this is facultative. So under certain growth conditions, very little oil. Under other growth conditions, including heterotrophic growth, so if we feed sugars to the algae, they'll produce very high concentrations of nonpolar lipids, and by that I mean triacylglycerol. So it's essentially vegetable oil. We can harvest all of the biomass. We're not leaving behind the roots and the leaves and the stems. It's 100% of the biomass is harvested. It's potentially year-round, 24/7. And because we're injecting CO2 into the ponds and hydrating that to bicarbonate, we're actually capturing the CO2 in the ponds before it could be emitted from point sources. And so we're reducing direct emissions into the atmosphere by injecting CO2 in ponds, and that turns out to be the substrate that it's actually moved into the cell for photosynthesis. Okay, so this is actually Chuck's Dismute slide, and co-author on this. And this slide shows the area in the US that would be required for growing corn to replace gasoline. And this is in the year 2006, and this is the actual corn crop area. And so you see for corn-based ethanol to replace gasoline in the United States is not entirely feasible, but it is feasible on a regional basis. So in the Midwest, where the majority of the corn crop is grown, it's a much more feasible technology than, say, perhaps in Maine. Cellulosics, which is getting a lot of attention and research, including here, has a greater potential and a greater feedstock base than is the corn cob. But if we look at various scenarios for growing algae, we see that the area required to replace all the gasoline is about the size of the state of New Mexico. We've approached the governor, but she's not agreed to cover the state in water yet. Anyway, so as I mentioned in the beginning, the NAB Consortium was funded in 2010. It involved 50 partners, including industries such as UOP on fuel conversion technology, Matrix Genetics on cyanobacteria metabolic flux analysis, small startup companies, a number of universities, the national labs, and independent research institutes. This really was a cradle-to-grave project, including algal biology, as I mentioned earlier, cultivation, harvesting, extraction, conversion, co-products and their role in impacting the economics of producing fuels, and then to keep us all honest in the sustainability and life cycle analysis teams. And this is one of our test bed sites. This is in Pecos, Texas. And we ran all of our outdoor trials actually in the southwestern portion of the United States. So I'm going to show you some data in a moment on various scenarios for producing biofuels and what are the technological hurdles that we need to address. But the base for that model is a farm of 4,850 hectares, as indicated here. About 4,000 of that actually in ponds. And you can see the volumes that we're dealing with. 330 days of operation were modeled. And you can see that the capital expenditure is huge. It's $1 billion to build a facility of this size. The operating over 5 years averages out close to that same number. So these are very large numbers. But it turns out they're very scalable as well. So the idea that you're going to grow fuels in your backyard swimming pool is really not feasible. It's not scalable at that level. So what I'm going to express in just a moment is a comparative analysis of the competing technologies. Technologies that existed in 2010 that were the baseline, and by and large are still the current baseline for technology introduction into algal biofuels production, and some newer technologies that came out of the survey that we carried out over the last 3 years of competing technologies. And so what I'm going to show you here are, first of all, some of the downstream technologies. And the reason for focusing initially on the downstream technologies, as you'll see in a moment, is one of the outcomes of the NAB consortium was the realization that the downstream technologies that are currently available and that were developed as part of the consortium actually meet all our requirements to be economically feasible. And they also meet our requirements to meet fuel standards, ASTM fuel standards, for fuels produced from bioalgae as a feedstock. The technology that we settled on as being the most efficient for fuel conversion is the two-stage process shown here, which is a combination of hydrothermal liquefaction and then the residual carbon that remains in the water phase from the hydrothermal treatment then is catalytically converted into methane. So we're producing both crude and methane. And then the nutrients, the inorganics that remain after the catalytic hydrogen gasification are then recycled back to the pond. And so that reduces the nutrient requirement for growing the algae. So this turned out to be the most cost-effective and efficient way of producing fuels. And here you can see the classes of molecules that are actually in this biocrude. But this process recovered 86% of the carbon in the biomass. Okay, so very little actually carried over as either CO2 or biochar. Okay, now one of the interesting outcomes, though, of analyses of fuels produced using the HTL process was the observation that very large differences in oil content of algae had a very minimal impact on the energy recovery in the biocrude. And this was not anticipated. So if we look at the relative energy recovery from various classes of biological molecules, so oil has an 87% recovery, starch about 14%, but here we're having a 6-fold difference in oil content, and yet we're only seeing a 30% difference in biocrude produced from these various algal fractions. So we don't know the reason for this at the present time, but it is an interesting observation because it turns out if you do a thermodynamic analysis of the energy conversion from solar energy into these various classes of molecules shown over here, and in particular focusing on oil and carbohydrates, it turns out that making carbohydrates biologically is about 10 to 20% more energy efficient than making oils. And there's a very simple reason for this. In the biochemistry of making oils, the substrate for making oils is acetyl-CoA, which is then polymerized to make fatty acids. Acetyl-CoA is derived from pyruvate. And pyruvate conversion to acetyl-CoA, you decarboxylate pyruvate, so you go from 3 carbons to 2. That's a carbon that was fixed through the process of photosynthesis. So every 2 carbons, you lose 1. That doesn't happen when you make starch. And so the energy cost for fixing that CO2 is conserved when you're making starch. So it raises the question, should we be making starch with algae, or should we be making oil? And if we're doing hydrothermal liquefaction, we're seeing that we're really not seeing much of a difference here with the 6-fold difference in oil content. Now, clearly there has to be a sweet spot here, but it's going to depend on the quality of the fuel that's going to be of interest to the combustion technologists. What kind of molecules do you want in that fuel? What is the optimal mix? And how do we tune the carbohydrate, protein, and oil ratios to give you that optimal configuration? So that's a linkage that we hope to develop with the combustion EFRC centers, is what is the optimal design of fuel or biological material to give you the best fuel composition? And then how do we tune that to give the highest thermodynamic efficiency for converting photons actually into carbon-carbon and carbon-hydrogen bonds? Okay, so this is the result of the technoeconomic analysis and life cycle analysis for 2 scenarios. The base scenario, which is the current technologies that are being used to produce fuels from algae, and the best case scenario that was resolved as a result of the research that was done through the NAB Consortium. And so you can see a variety of different technologies here, the biology and so on and so forth, and the relative biomass production. I should point out this is for the 4,000 hectare operation that was indicated in an earlier slide. And you can see that obviously by increasing the biomass productivity, in this case, this was achieved using genetic modifications. But if we had a 3-fold increase, we get roughly about a 3-fold increase in biomass productivity. And crude oil production actually goes up substantially, not only due to the GMO technology, but also the hydrothermal 2-stage process versus the wet solvent extraction, which is the current technology. The end products here are oil and methane versus oil and delipidated algae as a co-product that can be used for animal feed. And this product we actually tested in animal feeding trials, fish, shrimp, cows, pigs, and chickens. And it performed very well in nearly every case, as well as soybean meal. So if we go to scale and try to achieve levels of production that would displace gasoline in the United States, the amount of protein that would be produced is on the order of magnitude of current productions of protein from soybean and corn combined. So that's a tremendous amount of protein. Now, that raises the issue, are we actually competing for food? We would argue that we're replacing one food source with a different food source that can be used to feed animals. And so this would be a very massive amount of protein produced as a co-product by this process. Now, ultimately, it comes down to what's the price, of course. And current numbers that have been generated for the technology that's in place at the present time range from $16 to $230, so much higher than petroleum per gallon. With the best case scenario, we're starting to approach parity with petroleum. So this scenario here of genetically or enhanced production models, an arid raceway, which is essentially a river of algae flowing down a stream with an airlift technology at the front end, combined with hydrothermal and catalytic hydrogen gasification and the other factors that you see contributing here, is really the roadmap that's coming out of the NAB Consortium for how to go forward in the future. And the 2 factors that are going to have the largest impact are the ones outlined in red. That's biomass productivity, and so essentially photosynthesis, and harvesting of algae, how to do that more efficiently. Yeah?
The question that regarding that, the total cost. should be more narrowly defined,
Uh-huh.
not fitting in one order. With
Yeah.
future technology, it is uncertain what the technology will be, and
Yep.
that will be more broadly.
Yeah. So what I didn't mention is we put probability constraints around these numbers. And this is a 50% probability of occurring is the estimate for this particular, but it's the same number in this case as well. And these are literature numbers in large fractions, so yeah, there's a broader range there. Okay, so anyway, getting back to what are the constraints, it's biological production and also harvesting. Now, the electrocoagulation technology is essentially an electrode with a positive and negative surface that puts a charge on the, or neutralizes, I should say, the surface charge of the algae. They self-aggregate and they come out a solution. So a very simple, continuous flow process. It's actually been commercialized for sewage treatment facilities, and so it's clearly scalable to a very large level. That being said, it's very much dependent on the quality of the water and the chemical composition, particularly the ionic composition of the water. And so it can be challenging, and in an environment where algae are grown outdoors, where through evaporation and precipitation, the water quality is constantly changing, it could be a challenge. Now, to address that, we're actually developing biological approaches to auto-induce flocculation with good success. And we think that's a very low energy alternative to non-biological approaches to harvesting the cells. So that's in development and moving along nicely. All right, so the rest of what I'll be focusing on is some of the efforts that we've had underway over the last few years to improve the apparent efficiency of photosynthesis and to increase biomass productivity. And so photosynthesis, as you may know, can be sort of subdivided into two large groups of chemical reactions. Those that are known as the light reactions that are driven by light, and then the dark reactions, which can be run actually independently in the dark if the products of the light reaction, in this case ATP and NADPH, are provided. And so our research is focused on modeling the flux in these pathways, but also re-engineering these pathways to increase their efficiency. So this slide shows a breakdown of the energy utilization in photosynthesis. And if we start with 1,000 kilojoules of solar input, you can see how various energy losses occur across the initial steps, leading ultimately to the accumulation of biomass. And the bottom line here is we're looking at efficiencies that at best are 5 to 6% efficiency. Okay? Now, if we take apart this diagram, we can actually subdivide this into losses associated with light capture and energy conversion, and that's about half the energy loss. Energy conversion processes associated with the biochemistry of carbon fixation and its reduction to carbohydrates and oils. And then finally, the sink, the accumulation of that energy and storage products associated with, in this case, making corn, for example. Now, each of these stages, of course, is critical, and each of these stages interacts with the other stages. So there's feedback control from the source to the sink and vice versa, and so allosteric regulation plays a large impact on the overall efficiency. So largely until the last 10 years, the focus on improving photosynthetic efficiency has been on these latter two items: energy conversion, carbohydrate metabolism, and its accumulation. More recently, there's been a focus on light capture and energy conversion. Okay, so looking at the overall conversion of red photons into glucose, we estimate that its potential to achieve a 30% overall efficiency. based on the quantum requirement for photosynthesis and the energy content of glucose. So there's a 30% energy efficiency that's feasible using the current biochemistry of the cells. That's about 5-fold higher than the efficiency as it currently operates. And as I pointed out earlier, the energy return on investment for making carbohydrates is about 10 to 20% higher than it is for making oils, based on the loss of carbon that was previously fixed when converting pyruvate into acetyl-CoA. Well, the real problem with the light reactions is that the light harvesting apparatus that captures the light is actually much larger than it needs to be to saturate the downstream electron transfer processes. And so, as a result, if we look at the rate of photosynthesis as a function of light intensity, we see that photosynthesis light saturates around 500 micromoles of photons per meter squared per second, which is about 1/4 of full sunlight intensity, which is 2,000, roughly 2,000 micro-Einsteins. So these photons are being captured by the light capturing antenna complex, but they're not able to drive photochemistry because the downstream kinetics in electron transfer are much slower, and I'll show you this in a moment, than is the rate of light capture at full sunlight intensity. So those excess excited states then are dissipated as heat and fluorescence. And that accounts for a loss of 60 to 70% of the energy that it's actually impinging on the photosynthetic machinery that is photosynthetically active radiation. Okay, so what I'm showing you here is the electron transfer apparatus in the chloroplast of a plant, but also in the green algae that we work with. And it's made up of several major protein complexes that are shown here. This is called photosystem 2. It's also known as a reaction center, so it harvests light energy and it converts it into chemical energy. The source of the electrons that ultimately move through this electron transfer chain come from water. And so water is split, releasing oxygen and hydrogen. That's an area that Chuck has worked on extensively. And then it drives a linear electron transfer chain through a series of electron transfer carriers that also, in some cases, carry protons. So as the proton gradient is built up on this side of the membrane as a result of water oxidation and the transfer of protons across the membrane, those protons drive the ATP synthase machinery that makes the ATP. Then ultimately, the electron goes through one more charge separation stage driven by light in photosystem 1, and that leads to the reduction of NADP. So it's the NADP and the ATP that are used in the carbon-fixing reactions. Now, the reason I'm going through this in a lot of detail is what I've indicated here in these yellow boxes are the rate-limiting steps in these major complexes. So in photosystem 1, it's 1 microsecond. Here you see it's a millisecond and a nanosecond in this case. Light capture and migration of excited states through the light harvesting apparatus is a quantum coherent process. It's very efficient at room temperature and very fast. So the antenna complexes, which are shown out here in the light green, where the light is actually captured and funneled into the reaction center, are very, very efficient, whereas the rate-limiting step here is 1 millisecond. So as I pointed out earlier, at full sunlight, the rate of capture actually exceeds this rate limitation by a factor of 10. So that's the bottleneck. Okay. So about 15 years ago, Tassio Mellis at Berkeley made the observation that if he decreased the size of the antenna complexes, which are shown here, surrounding the photosystem 2 apparatus, shown here, by reducing the synthesis of one of the pigments that is uniquely associated with the antenna complex, that the rate of photosynthesis actually increased as a function of light intensity. And so what he did was he identified a mutant that could not make chlorophyll B. So there are two types of chlorophyll in plants and in the green algae, chlorophyll A and B. And A is associated with the reaction center complex and is involved in the charge separation. Both A and B are found in the antenna complexes that are on the periphery of this reaction center complex. And so elimination of chlorophyll B prevents the translational assembly of the light harvesting complex, and so you can diminish it substantially. Now, in this case, they eliminated all the chlorophyll B. And so these complexes, which are called light harvesting complex trimers, these monomers shown here that are all part of the antenna complex and account for 80% of the chlorophyll, were all lost in this particular mutant line. And only the chlorophyll associated with the proximal antenna that is part of the reaction center complex remained. So we go from about 250 or 500 chlorophylls, excuse me, down to about 80 total. And so that's a substantial reduction in the light harvesting capability. Okay, so that was back around 2000. And here we go. This is a picture that shows a little bit more detailed organization of these reaction center complexes with their antennas. And I'm not going to really dwell on this very much, other than to make one point, and that is various different members of the antenna complex have different ratios of chlorophyll A to B. And so the trimers have the highest amount of chlorophyll B, it's 1-to-1 ratio. And then some of the smaller subunits have much higher amounts of chlorophyll A than chlorophyll B. That turns out to be critically important. So these complexes, these small antenna monomers that are very closely associated with the reaction center and very tightly coupled to the reaction center, have the lowest levels of chlorophyll B. And so if we were to generate a gradient of reduction of chlorophyll B, the expectation is the trimers would disappear first, and then that would be followed by the very tightly associated monomers. And I'm not going to show you the data in this talk, but we've actually done this analysis by mass spectroscopy, and that turns out to be the case. So elimination, gradual elimination of chlorophyll B eliminates the most loosely associated complexes, and then eventually that's followed by the tightly associated complex. So that has implications actually for the kinetics of energy transfer and the efficiency of energy transfer. So if the tightly remaining complexes persist as we reduce the amount of chlorophyll B, we actually see that the quantum efficiency at low light intensities increases rather than decreases, because we're not moving energy captured by these LHCs that are loosely associated, long-distance energy transfer, through into the reaction center. We're only getting energy transfer from the tightly associated complexes. This was an unanticipated result initially, but it's an exciting result because what it tells us is we reduce the size of the antenna, it actually gets more efficient in energy transfer at low light. And low light conditions in a gradient of light, for example, in a leaf or in a pond, are a reality that we have to deal with. Okay, so the antenna actually has multiple functions. It not only harvests light that can be provided to drive electron transfer, but it turns out that the antennas can move between the two photosystems laterally in the membrane and redistribute energy between the two photosystems so that if they get out of balance, they can rebalance electron transfer between the two photosystems. And this process is called state transitions. Very well understood and very well modeled. So energy balance is one of the roles of the antenna. There's another function associated with that, and that is the antennas are generally most closely associated with photosystem 2, but as they move to photosystem 1, they allow for a process called cyclic photophosphorylation to occur. And I'll show you a picture that describes that in a moment. But the bottom line is it allows for the plant or the algae to make additional ATP beyond that that's generated by linear electron transfer. And that turns out to be very important for the algae. The algae use ATP to drive the uptake, active import of bicarbonate into the cell. So this protein, which is in the plasma membrane, the outside of the cell, uses ATP, it's an ABC transporter, to move bicarbonate into the cell. Ultimately, it moves through another transporter into the site where the CO2 fixation occurs. So algae require an additional ATP for every CO2 fixed, above that required by low-efficiency photosynthesis plants. So to meet that demand, they have to be able to make more ATP, and it turns out that in algae, they have a very active, what's called cyclic electron transfer system, unlike low-efficiency plants, which have a very inefficient cyclic electron transfer. So in cyclic electron transfer, the electron essentially travels across the membrane back and forth, and as it does so, it facilitates the movement of protons across the membrane that drives ATP synthesis. So you can imagine if we move the antenna complex from photosystem 2 to photosystem 1, we're increasing the optical cross-section of photosystem 1, which drives this reaction, and that can facilitate additional ATP synthesis. Now, the reason I'm mentioning these additional functions of the antenna is that if you'll recall, the original mutant had no peripheral antenna. So that would mean there would be an impairment, potentially in cyclic electron flow, that's rebalancing electron flow between the photosystems would be blocked. And it turns out there's another function associated with the antenna that's associated with quenching excess energy, which I'm not going to describe, but we've actually published on this. And that process is associated with the antenna complex. So that would be lost if we lost all of the antenna complex, at least the peripheral antenna. So that led to the hypothesis that the solution to all problems is moderation and not excess going one direction or another. And the idea then was that an intermediate size antenna may actually be optimal for photosynthesis and increase its efficiency. So the genetic approach that we took was to rheostat and regulate the synthesis of chlorophyll B by moderating the expression of the enzyme that converts chlorophyll A into chlorophyll B called chlorophyll A oxidase. So we use small interfering RNAs or siRNA technology to downregulate the expression of this and to generate a series of transgenic organisms that had a range of antenna size. So the objective is really to move away from a model that looks like this. So if we look at a tree canopy going from the top to the bottom, every leaf has roughly about the same antenna size. Or if we're thinking about a pond where we're growing algae, we'd like to move away from a scenario like this to a scenario like this, where we have smaller antenna at the surface of a tree or at high light intensities in a pond, and larger antenna at the base to capture the remaining photons and to drive higher rates of photosynthesis. So that's the objective that we undertook. So this shows the chlorophyll A-B ratios of a variety of transgenic organisms. Here is the wild type, so it has a lot of chlorophyll B, so the ratio is 2. And you can see we generated a range of A-B ratios, and of course we have a mutant that makes no chlorophyll B. This is the mutant from the Berkeley group that I mentioned earlier. So to get an idea whether the A-B ratio was a proxy for the antenna size, we looked at rapid chlorophyll fluorescence rise kinetics and showed a strong correlation between the rise kinetics and the chlorophyll A-B ratio, as you can see here. So the A-B ratio essentially serves as a proxy then for the antenna size in this range of transgenic organisms. But we can also show that the antenna size is moderated in size biochemically, and that's shown here in a polyacrylamide gel under native conditions. This particular protein band that you see here that's green is the extrinsic light harvesting apparatus. And you can see in the mutant with no chlorophyll B, it's entirely missing. as was shown earlier. The intermediates, on the other hand, with higher AB ratios, have intermediate amounts of this complex. And we can also show that by fluorescence intensity from the chlorophyll on colonies growing on Petri dishes. So all of this shows that we can modulate the size of the antenna. And then, of course, the question is how do they respond photosynthetically for CO2 fixation as a function of light intensity? And so here's wild type in the dark green, and that saturates, again, around 500 micromoles. The small antenna, and this is all CO2-dependent photosynthesis, is doing better, but not nearly as well as published data. But I do want to point out the published data was actually photoheterotrophic growth. So acetate was in the media as an alternative carbon source. This is entirely photosynthetic growth. The surprising thing was nobody ever actually measured it based only on photosynthesis until more recently. These are the intermediates, and they're doing quite well, as you can see. So we're getting about a 2.5-fold increase in photosynthetic rate. So does that translate into biomass? And so this is the increase in biomass under low light conditions. So 50 micromoles, that's about the light intensity in this room. And 500 micromoles are saturating light intensity. So the intermediates do just as well as wild type. And that we were very pleased with that observation. This, of course, is the very small antenna mutant, and it does very poorly at high light intensities. But low light intensities. But at high light intensities, the intermediates actually do better than wild type. And here we can see that the growth rate is actually slower than wild type for the very, very small antenna. And this had not been published previously. So the secret behind the increase of photosynthesis is it doesn't necessarily correlate with the growth rate. Okay, so that technology demonstrated that an intermediate size antenna was probably optimal. And it turns out, we found out that a chlorophyll AB ratio of around 5 to 6 was the optimal AB ratio for photosynthesis and also for biomass productivity. Now, of course, during the course of the year, and actually on a daily basis, light intensities change dramatically. And they'll change in a canopy as light moves through the canopy with sunflex. So ideally, if we want to increase the efficiency even further, we'd like an antenna system that could rapidly self-adjust to different light growth conditions. So in the winter, we'd want a larger antenna, and in the summer, a smaller antenna, or at high light intensities. And so we developed a gene expression technology that would actually self-adjust the antenna size as a function of light intensity. And we did this in algae, and to do this, to build this system in, we had to start out with the mutant that couldn't make any chlorophyll A. Then we put the gene back in for chlorophyll A oxygenase. But at the 5' end of the messenger RNA, we fused what's called a light-responsive element. This light-responsive element will interact with a protein called a nucleic acid binding protein 1, or NAB1. And when NAB1 binds to the light-responsive element, it prevents the translation of this messenger RNA into a protein. So this is translational regulation, not transcriptional regulation. Now, importantly, under high light conditions, naturally in the algae, NAB1 levels are high, and under low light conditions, they're low. So under high light conditions, we get translational inhibition of conversion of chlorophyll A into B. The AB ratio, therefore, goes up. The antenna size goes down, as we would desire. And the prediction is the culture productivities under high light will increase. Under low light conditions, there's very little expression of NAB1. So we get good translation of this gene construct. The antenna size gets larger, and the productivities are predicted to go up under low light conditions. So these are our artificial ponds. These were built as part of the NAB Consortium. They mimic ponds in depth and in every environmental parameter: light intensity, light duration, temperature, pH control, stirring, etc. We can mimic any environment in the world with these so-called environmental photobioreactors. And this company was actually a spin-out of the NAB Consortium program. So under these conditions, we mimicked a day in New Mexico in the spring, but with full sunlight intensity of 2,000 microeinsteins at noon. And under those conditions, if we follow the chlorophyll A-B ratio, as the culture density increases, the antenna size gets larger. And you can see that in the transgenics here by following the chlorophyll A-B ratio. Wild type, no change at all. If we look at the photosynthetic rates as a function of light intensity for a variety of different transgenics as shown here, the transgenics that actually had the highest chlorophyll A-B ratio gave us the highest photosynthetic rates. And again, those chlorophyll A-B ratios were in the range of about 5 to 6. And here you can see we're getting a 3-fold increase in the rate of photosynthesis. So this is one of the greatest increases in photosynthetic rates that's been achieved to date. If we look at the productivity, we also see a very similar relationship. So now we're getting up to a 2-fold increase in biomass productivity by engineering a self-adjusting antenna, and that's this. This is culture density shown here, but this is actual dry weight, and this is a variety of various controls. The mutant missing chlorophyll that does not make chlorophyll B, and that same mutant complemented to restore that capability, but without the self-regulating antenna adjustments. And so clearly a 2-fold increase. So as I mentioned earlier, one of the outcomes of the technoeconomic analysis of the NAB Consortium was the requirement to have at least a 3-fold increase in biomass productivity. So we're 2/3 of the way there, we think. And these organisms will soon be going into trials in the outdoors. Now, one of the advantages at being at a national laboratory such as Los Alamos is there is a lot of additional big science capability at Los Alamos. And one of the areas that we are particularly attracted to was the development of giant quantum dots. And these are quantum dots that are made with non-toxic heavy metals that are about 30 nanometers in size, have a very large Stokes shift, and essentially don't blink. They're robust, they can operate over multiple years, and they're very insensitive to heat inactivation. So much superior to past technologies for quantum dots. So this is not the surface of the sun and sunspots. What you're seeing here are microencapsulated algae in a hydrogel. And this hydrogel has been designed with a particular porosity that we're able to measure by small angle neutron scattering that facilitates the diffusion of gases in and out of the gel, but also nutrients. Embedded in the hydrogel are the giant quantum dots. And the objective of integrating the giant quantum dots is to frequency shift green photons, which are not used by photosynthesis, to red photons. We're increasing the photon flux into these algae, but keep in mind with normal wild-type algae, of course, that would not give you any advantage because photosynthesis is presumably already light-saturated. But with the self-adjusting antenna algae, then the idea is that the algae on the exterior of this bead would have smaller antenna, and those toward the interior would have larger antenna, but they would self-adjust in response to the photon flux density. So as we increase the photon flux density by frequency shifting green light into red light, we're actually increasing the number of usable photons to drive photosynthesis. So the first stage in testing out this model was to actually see if algae would grow well in these encapsulated hydrogels. And the answer is they actually grow better than they do in liquid solution. So we actually, and this is the same algae that we worked with in the antenna system, we got a 3-fold increase in productivity based on measuring chlorophyll as a proxy by just encapsulating the algae in the gels. At this point, we don't know what that's attributed to, but that's an area that we're investigating. Now, to see if we could actually get energy transfer from the giant quantum dots to the algae, What we did was grow up these beads with the giant quantum dots and algae over a period of time. And using various fluorescence filters, we can actually discriminate between fluorescence attributed to the giant quantum dots, which is shown here in red, and chlorophyll fluorescence that's indicated in yellow. And so what you see here is initially the fluorescence is essentially all emerging from the giant quantum dots, but as the algae colonies grow, and this is falsely colored red here, so it's not the same wavelength as here, but these are actually colonies of algae growing in these beads, and we see this transition between intermediate levels of fluorescence from the quantum dots. Here it's shown in yellow, but essentially at the end it disappears. So we're capturing the energy from the quantum dots by the algae to drive additional photosynthesis. So where we're going with this now is actually testing this with the antenna complexes that can self-adjust. So the last part that I want to mention very quickly is the work that we're doing in higher plants. And can we translate these technologies from algae into higher plant systems? Because the bottom line, of course, is most of our biomass is coming from plant systems. So we've taken the same approach. We're working with a plant called camelina, where we're down-regulating the size of the antenna. And here you can see a transgenic camelina coming up in a population of wild type that's growing much faster. So we use an RNAi-based approach, and the targets were various elements of the chlorophyll A oxygenase gene, and we had different RNA constructs, a short one and a long one. And I'll not mention any more beyond that. So here you can see the clear color differences between a wild-type camelina plant and one with an engineered antenna that's about the optimal chlorophyll A-B ratio. We can also demonstrate a reduction in the antenna size by looking at the chlorophyll fluorescence emission. So here's the transgenic at top. The red is higher fluorescence levels, and you can see much reduced fluorescence in this case. And eventually we get quenching of the fluorescence by de-excitation pathways in the wild type. And we see evidence for that occurring also in the transgenics. If we look at photosynthetic rates, here we have a range of antenna size modulated by the chlorophyll A-B ratio ranging from 12, very small antenna here, wild type at 3, and these are intermediates that are 5.4 and 7 for the chlorophyll A-B ratios. So looking at the rate of photosynthesis as a function of light intensity, we're getting about a 25% increase. So these are the full-grown plants. The seed pods are harvested to produce oil. This is a plant very closely related to canola. And you can see in general that they're somewhat larger, but another interesting feature is the persistence of the more mature leaves longer on these plants than you see in the wild type. And this we see over and over again. So we have more photosynthetically active surface area in these plants that have the intermediate size antenna than we observe in the wild type. If we grow them side by side in soil as they're growing outside, here you can see the transgenics are larger than the wild type, and so that's encouraging. And with that, I'll summarize. So where we stand to date is we've demonstrated through the NAB Consortium that the pathway to making algal biofuels economically feasible is an increase in biomass productivity, a reduction in the harvesting costs, in combination with the leading technologies for fuel conversion, which at this point are hydrothermal liquefaction and catalytic hydrogen gasification. We've shown that algae with self-adjusting antenna have twice the biomass productivity. So we're 2/3 of the way of meeting our objective of a 3-fold increase in productivity. We've also, and I haven't shown you this data, but it turns out that having large antenna is actually evolutionarily more fit than having a smaller, more efficient antenna. And it comes down to competition for light. So even if you're wasteful in harvesting the light, but you don't allow that light to be available for competing species, different species, then you win in the resource competition. And so in our studies, when we compete large antenna versus small antenna, the large antenna organisms always win, and by a substantial amount. That has implications for how we actually grow the algae with self-adjusting antenna in the pond, and I can talk about that later if you'd like. We've also found that antenna size is optimal antenna size is a chlorophyll AB ratio around 5 to 6, and it's true for both plants and for the green algae. Now, green algae and plants, it turns out, have very similar antenna complexes and designs, and so it's not unexpected that they would have similar values. And finally, we're developing biohybrid devices that I described briefly that combine solid-state technologies with hydrogels to encapsulate algae, increase the number of available photons to dry photosynthesis, but also allow us to more efficiently harvest the algae potentially using nets or gravity-based systems without a tremendous energy input. And so thank you for your attention, and happy to take questions. Yeah.
In one of the earlier slides, you talked about the kinetics of the process and using the timestamps.
Yeah.
And I forget which picture it was. One of them had a timestamp of 1 millisecond.
Yes.
Can you just reiterate your question to the microphone? So we're recording it, so it's good to have the question.
Okay. So in one of the previous slides, you had a listing of the timestamps for the kinetics of each of the different systems where you take light and you harvest it, and then you go through photosystem 2 and 1 and whatnot. And I forget which picture had it, but one of them had 1 millisecond.
Yes.
But and that seemed to be the rate limiting step.
Yes.
If I'm correct. But then the rest of the presentation seemed to look into changing the original light harvesting antenna so that you can basically almost slow down that process so that you can have that all. Why not optimize that 1 millisecond so you can make it into a much smaller time?
Yes.
So that you can make everything else, and then you wouldn't have to deal with competition in terms of the antennae.
Yes, and so we're trying to do that. It's particularly challenging. This cytochrome B6F complex oxidizes reduced plastohydroquinone, or plastoquinol, excuse me. And the binding site where that reaction takes place involves a protein hinge that moves in and out, that is kinetically very, very slow. And it involves an electron loop called the Q cycle that was actually the basis of a Nobel Prize many years ago. But anyway, that engineering that hinge is particularly challenging. So one approach that we're looking at is actually increasing the pool size of the acceptor molecule, plastoquinone, to buffer transients in electron flow rates generated from photosystem 2. And there's actually evidence in the literature that suggests that this can work, but only to a certain extent. And there are two parts to this story. One part is it provides a buffer for fast and slow electron transfer processes. But the other issue, it turns out, is this plastoquinone molecule can actually quench chlorophyll excited states. And so effectively reduce the antenna size, but in an indirect way. And we've shown evidence in some of our engineered organisms that that's a contributing factor to the increase in photosynthetic rate. Now, overall, we still haven't gotten there. This antenna strategy is much superior. But there's another approach, and that is to increase the abundance of these B6F complexes, to change the apparent KCAT for overall electron transfer. So normally the ratio of photosystem 1 to this B6F complex to photosystem 2 is 1 to 1 to 1. And so then we're limited by that slow step. But if we had 10 times more B6F complex, then potentially we could increase the apparent Kcat by even more. We're looking at that approach. We haven't identified the gene that regulates the abundance of that complex, but that's part of the approach. So we are certainly looking at that. Thank you.
From your talk, would it be right to say that the lipid production in algae doesn't seem to matter that much at all? It could be any organism that produces quick biomass would work in the process you showed. So is that correct to say that?
Let me say that biofuel production from biomass is becoming more and more feedstock agnostic. That means that cellulose may be as efficient as algae. Now, with many caveats, of course, oil, much more reduced, more hydrogen. That has implications for the end products that come out in the biocrude, and tuning that biocrude so that it's optimal for refineries and for combustion purposes is one of the objectives that we want to focus on in the future. And that's where we think we can particularly interact with the combustion group here, is identifying what's the best mix of protein, carbohydrate, and lipid to give you the best feedstock for combustion purposes. So there's lots of room for wiggle in there. But the surprising thing, as you noticed, is that a six-fold increase in oil doesn't mean a six-fold increase in energy return.
Very nice talk. I have a couple of questions. The first one is that a lot of the effort that you are putting in is try to suppress the amount of chlorophyll B that you're forming in the antenna. I'm just curious as to why did nature evolve to making the nature chlorophyll B in the first place? Kind of a biology question.
Okay.
The second question is you showed some numbers about the capital cost for a 4,000 hectare unit, about $1 billion.
Yeah.
I'm just imagining what would happen if I scaled it up to New Mexico, which is what you wanted, right? Ultimately.
Yeah.
So looking at a few trillion
so
dollars
$500
there.
billion
$500 billion?
is roughly the estimate. And why chlorophyll B? Chlorophyll B absorption spectrum is somewhat blue shifted to chlorophyll A, so it increases the number of photons that can be captured from the solar spectrum. Chuck.
I have a couple questions too.
Yeah.
With regards to the practical aspects, there's a lot of effort to try and use captured CO2 from flue gas, and so one potentially could, if one had a smart organism, use the NOx and the sulfur as well as a nutrient component. Plus, obviously, the CO2 is of some benefit. So that's one of the challenges that presumably would get around having to do all this fancy genetic engineering. Is that a practical solution as well?
So there have been studies that have compared the growth rates of algae using pure 100% CO2 versus flue gas. And in most of those studies, algae do better with flue gas. So it's apparently, one, not toxic, and those additional nutrients have some value. So there is an upside to flue gas. Now, can we improve upon that? That actually raises another issue that I didn't talk about that's part of the NAB consortium. One of the big challenges in growing large-scale agriculture usually is, of course, herbivory and pathogens. And when we go to that scale, it's going to be ripe territory for all of those problems. And herbivores and pathogens, we've found, can crash ponds very rapidly in a matter of a day or two, go to zero productivity. One of the strategies that we've identified as a way to control that problem is what we call nutrient pulse technology. Algae, as you know, grow in essentially nutrient deserts in the open ocean. There's no iron to speak of. There's very little nitrogen available. And what we've learned is, through molecular approaches, is they have very active nutrient transporters and storage capabilities in the vacuole. And we've identified algae that we can preload with nutrients and let them grow a week in distilled water, and they grow at rates unimpeded relative to nutrient-rich water. So sort of roundabout coming back to the nutrient issue, we think one of the strategies to control contaminants is to preload the algae in an inoculum with nutrients and put them essentially into distilled water. which would not have nutrients available for competing organisms. And that would diminish the problems with herbivory and pathogens. Yeah.
So if I can ask the other question,
Yeah.
one of the other, well, the other requirement is for growth at temperature. So
Yeah.
when you go to the third world, they're going to be using open ponds, and they're going to be going to high density likely, because they don't want to move too much water all the time. So you end up with temperatures that are well above ambient, and so you end up with obviously stress and death and pond crashes and so forth. So is there a strategy for overcoming this problem?
Yeah. And it's more than just growing it in developing countries where it's going to be hot. One of our major concerns with algae, of course, is the water requirement for growing the algae. And currently with open ponds, which is the only feasible technology really for growing algae, forget closed photobioreactors on this scale. It's too expensive and they foul. And the cost of cleaning them is again too high. That was built into the LCA models, by the way. So we're going to have open ponds and we want to reduce water loss. So the cheapest way to do that is to cover it with plastic film, and that'll substantially reduce evaporative cooling, water losses, but the temperatures will go even higher. A company called Algae in Florida is now doing exactly this in closed systems, essentially plastic bubbles with plastic-contained algae. And they've observed in Florida that the maximum temperature is 55 degrees centigrade in the summer with no evaporative cooling. So that's the benchmark that we're concerned about right now. So we're pursuing two strategies to deal with the temperature stress. One is extremophile algae that can grow at very high temperatures and grow very fast. We've identified one that looks very promising. It produces high biomass at 55 C, so it hits our target. And it grows at very low pH, so it's going to pH 2, and so competition from other organisms would presumably be diminished there as well. So that's our summer algae. The winter algae and spring and fall algae, we've identified through our bioprospecting efforts at NAB in a screen of 2,000 wild isolates, a chlorella strain that grows well at 37 degrees. That's its optimal temperature, which is quite warm. And we're now doing directed evolution approaches to bump that up to about 45 C. So if we get to 45 C, we've calculated that that algae in combination with the thermophile will give us the best model for year-long production. So thermophile in the summer, 45-degree maximum temperature chlorella strain throughout the rest of the year. So that's sort of a two-pronged approach to addressing the issue of temperature. Then we cover the ponds and we probably eliminate evaporative losses by 90%, something on that order. Yeah.
What about nanochlorosis?
Yeah, I don't like them. Sorry, I'm being blunt. Well, yeah, they did. Nanochloropsis, as you know, grows in seawater. If you're not near the ocean, you're going to have a problem with evaporative water loss in the current technologies that's going to concentrate the salts in that pond. And the only way of addressing that is to take that water when it reaches a point where the algae can't grow anymore, moving it to a blow-off pond and drying it down to salt. Or alternatively, supplementing with fresh water. Neither of those strategies is economically feasible. We've looked at that through LCA analysis. It will not work. Chevron figured this out 20 years ago. When they first started looking at algae, they said, do not start with marine algae in the continental United States. It's a non-starter. The only place we think where that's really going to work well is near the coast where you can exchange seawater in and out. But we're not fans of marine algae in areas where you can't exchange the water at all. Yeah.
One more question.
Yeah.
Hi, thank you very much for the interesting talk. The biofuel, like algae-based biofuel, has climate benefits in terms of replacing the fossil fuels and also uptake some of the carbon dioxide. But it might also have the negative impact in terms of emitting, say, methane or nitrous oxide into the atmosphere, which are much stronger greenhouse gases. So I just wonder, like, during your research, whether you also look at that aspect?
So the answer is no. We haven't looked at methane or nitrous oxide emissions from algae. NO is a possibility, but I just don't know the magnitude of what that would likely be. But based on what we know about the biochemistry of these organisms and their one-carbon metabolism, we would not anticipate any methane emissions. So, yeah. But that's a good point. It's something to look at. Yeah. 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 Certainly. Yeah. Yeah. Okay. Yeah. Something to look at. Yeah. Thank you. Yeah. Okay.
All right, before we wrap up, I get the last word, being from Iowa. So my question would be, how long, if you get the $500 billion, how long until you can build the state of New Mexico covered in algae?
Yeah. So if you'd asked me that 2 years ago, I would have said, yeah, we can do it. It's all going to happen. But the reality of the world has changed. Probably never changed. My reality changed. But I've been involved in a startup company that's trying to commercialize algal biofuels. And the challenges have been government regulation and changing policy on carbon emissions and renewable fuel standards. And the change in the recent change in November in the renewable fuel standards for advanced biofuels versus corn-based ethanol and elevating the corn-based ethanol standard did very big damage to the companies that are startups in the emerging advanced biofuels arena, because now the demand is not there. And so that's hurt the industry. But the other bigger challenge is moving from the $100 million level to that $1 billion or $500 million level. That takes a level of investment that can only be provided by very large financial institutions like Morgan Stanley and Lehman Brothers and so on. And we and others have approached those investment banks. It's a non-go because they're so risk adverse now, they're not willing to invest in any technology that's not already profitable. So no R, all D, and algae's not there yet. So there's the so-called valley of death. That's anything above 100 million. It's not going to happen. So I don't see algae biofuels happening unless the government puts in place a carbon tax that makes it advantageous to go in that direction to sequester carbon. And without that, the industry is not going to move in that direction. So it's dead in the water until that happens. Yeah.
Well, we'll end on that great pun. And if you have more questions, come down. But thank you again to our speaker.